Hello dear student. Today we are going to discuss the topic immunoglobulin. It is also known as the antibodies. The objectives of today's topic to understand general structure of immunoglobulin, types of immunoglobulins, structural variations of different immunoglobulins, function of different immunoglobulins, clinical significance of that particular type of the immunoglobulin. What exactly the immune response? Immune response is collective and coordinated response to the introduction of foreign substance in an individual, mediated by cells and molecules of the immune system. Means humoral as well as cellular immunity works harmoniously and then will get the immune response after the entry of the particular antigen in your body. Immunoglobulins derive their name from the finding that they migrate in the region of globulin when antibody containing serum is placed in an electrical field. Immunoglobulins are glycoprotein molecules, function as antibodies worked by plasma cells in response to the immunogen, means that is the antigen. Structural characteristics, heavy chain and light chain. The immunoglobulin is having a 4 chain, 2 heavy chain and 2 light chain. Then disulfide bond is present in the immunoglobulin. The heavy chain and light chain and 2 heavy chains are held together by interchain disulfide bond and non-covalent interaction. Interchain means the disulfide bond between heavy chain and light chain, while intrachain disulfide bond is the bond within that chain. Means this disulfide bond is present within the heavy chain, within the light chain. It is known as the intrachain disulfide bond. Then these antibodies are also consist of the variable region and constant region. Based on the variability in the amino acid sequence, both the chain are having a variable as well as the constant region. The number of amino acids present in that region is vary. This region at which arms of the antibody molecule form a Y, means this region, middle part of the Y, is called as the hinge region because it is the flexible. Then folded into a globular region of each constant, which contain an intrachain disulfide bond is called as the hinge region. It's called as the domain, and this domain is globular structure and forms the light chain domain and heavy chain domain. Then next part is the carbohydrate, which is attached to the CH2 domain in most of the immunoglobulin, and it may also be attached to another region depending upon the type of the immunoglobulin. Then immunoglobulin fragments after the digestion of the proteolytic enzyme, when the immunoglobulins treated with the different types of the enzyme, for example papain, they are breaking down into the small part or fragment immunoglobulin molecules in a hinge region before hydrogen-hydrogen interchain disulfide bond. When it treated with the papain, there is a breakdown in the hinge region and upper and lower part get separated. This region is known as the Fab region because B stands for the antigen binding site and it is known as the Fab region. Then remaining part is the FC region. FC region is the region because it is easily crystallized, that's why it is known as the FC region. It is the remainder part of the heavy chain containing CH2 and CH3 domain. Then structure function relationship. Antigen binding function of the immunoglobulin is carried out by Fab part. Means in that Fab part antigen get bind. Then next the effector function. Effector function are mediated by Fc part of the molecule. Different functions are mediated by different domains in this fragment. Then FAB2. What exactly is FAB2? When your immunoglobulin is treated with the pepsin, in the cleavage of heavy chain after hydrogen-hydrogen interchain disulfide bond, resulting in the fragment that contain both antigen binding site. This fragment is called FAB2 because it is divalent. It is having two sides for the binding of the molecules. That's why the region is known as the FAB2. Then general functions of the immunoglobulin. Antigen binding by antibody is a primary function and can result in a protection of the host. Each immunoglobulin binds to the specific antigenic determinant. Valency of antibody refers to the number of antigenic determinants that an individual antibody molecule can bind. Means if the antibody can able to bind with the two antigens, then its valency is 2. If it is capable of bind with the 4 antigen, then its valency is 4. Next, effector function. Effector function includes complement fixation, binding to various cell types, placental transfer, and opsonization. Then general functions of this immunoglobulin: binding to various cell types of, say for example, phagocytic cell, lymphocyte, platelet, mast cell, basophil, and have a receptor that bind the immunoglobulin. This binding activate the cells to perform some functions. And then some types of the immunoglobulin also bind to the receptor on the placental trophoblast and transfer to the fetus. As a result, maternal antibodies provide the immunity to fetus and newborn. Then classes of the immunoglobulin. Based on differences in the amino acid sequence in a constant region of heavy chain, there are 5 subclasses: gamma heavy chain, mu heavy chain, then alpha heavy chain, delta heavy chain, and epsilon heavy chain. While based on the amino acid sequence in a constant region of light chain, 2 subclasses are there: kappa light chain and lambda light chain. Then structure of the IgG immunoglobulin. Subclasses of these IgGs depend upon the disulfide bond, length of the hinge region. In properties, it is the major antibody in the serum, major antibody in extravascular spaces, only immunoglobulin that crosses the placenta, complement fixation and opsonization. Then functions, secreted in high quantity in secondary exposure, means secondary exposure of the same antigen, then it crosses the placenta. Major functions, neutralize the microbes, opsonize antigen, activate complement and protect the newborn. Next type of the immunoglobulin, that is IgM. Okay, it is pentamer, but it can exist as a monomer. All heavy chains are identical and all light chains are identical. Valency is 10 because pentamer is there and each unit is having a valency 2. That's why this valency of the IgM is 10. Extra domain on the mu chain, and it has another protein covalent bond via disulfide bond called J chain. Means if you see this orange color circular structure, which is responsible for the binding of the 5 subunits together, and it is known as the J chain. This chain function is polymerization of molecules into pentamer. Then properties, third most common serum immunoglobulin, first immunoglobulin to be made by fetus, a good complement fixing immunoglobulin, very efficient in leading to lysis of the microorganism. Then good agglutinating immunoglobulin, cellular binding of IgM to some cell via Fc receptor. Then beta cell surface immunoglobulin, surface IgM exists as a monomer and lacks the J chain, but it has an extra 20 amino acid in C terminal to anchor it into a membrane. Cell surface of IgM functions as a receptor for antigen on B cell. IgM initially secreted during the primary infection. It cannot cross the placenta. Secreted first during the primary exposure, activate the complement, used as the marker of recent infection. Then IgA is a monomer found in secretions in the dimer. In a dimer, J chain is associated with the dimeric form. Then secretory piece is responsible for the binding of the 2 subunits. Secretory IgA is sometimes referred as the 11S immunoglobulin. Okay, means the secretory piece is responsible for the binding of the 2 subunits. Then IgA property and function. It is the second most common serum immunoglobulin. IgA is a major class of immunoglobulin in secretion. For example, tears, saliva, then mucus. Normally, IgA does not fix the complement unless aggregated. IgA can bind some cells like lymphocytes, dimeric with secretory component in the lumen of GIT and respiratory tract. Then major functions, neutralize microbes and toxins, Serodiagnosis of tuberculosis. Structure and functions of IgD. It exists only as a monomer found in a low level in serum. Its role in serum is uncertain. IgD is primarily found in beta cell surface where it functions as a receptor for antigen. Immunoglobulin D does not bind complement. IgE exists as a monomer and has an extra domain in the constant region. Then properties of IgE. It is least common since bind very tightly to Fc receptor on basophil and mast cells even before interacting with the antigen. IgE does not fix complement involved in an allergic reaction. It also plays a role in parasitic helminth disease. Serum IgE level rise in parasitic disease and measuring IgE level helpful in a diagnosis of the parasitic infection. Then eosinophil have a Fc receptor for IgE and binding of eosinophil to IgE coated helminths results in killing of the parasite. Associated with anaphylaxis play role in immunity to helminthic parasite, example allergic, bronchopulmonary, aspergillosis, parasitic disease. Thank you so much for patiently listening.